Good afternoon, ladies and gentlemen. I'm Jessica Shantz, and this is your afternoon news report.
And I'm Madison Smart, here to talk about the newest breakthrough in research for Alzheimer's and ALS.
ALS, or amyotrophic lateral sclerosis, is a progressive neurodegenerative disease that affects nerve cells in the brain and the spinal cord. In the nervous system, most motor neuron cell bodies are located in the spinal cord. A signal is then sent to the cell's axon, which sends the signal to a muscle receptor. This disease works against that process, however. In this disease, the motor neurons break down and die, and therefore they are unable to communicate with the rest of the body. When the brain and spinal cord are unable to communicate with the body's muscles, the muscles become very weak. This is called atrophy. Over time, a person with ALS will no longer be able to control their muscles because the brain and spinal cord aren't able to send messages to different parts of the body. This may result in a person being unable to do many everyday tasks. Without the communication between the brain, spinal cord, and muscles, a person wouldn't even be able to breathe on their own. The diaphragm, the muscle that controls the inhale and exhale of air in vertebrates, requires signals from the brain to operate. Without communication from the brain, the ALS patient wouldn't be able to breathe on their own.
A similar disease to ALS is Alzheimer's. These diseases are similar because they are both neurodegenerative disorders that result in loss of body function. Alzheimer's is an irreversible, progressive brain disorder that slowly destroys memory and thinking skills. The main cause and or feature of Alzheimer's is the loss of connections between nerve cells in the brain and eventually the death of nerve cells. The death of these nerve cells is caused by the misfolded proteins amyloid beta, which creates a buildup of plaque and tau, which creates neurofibrillary tangles. Since 2003, there have been no new FDA-approved ways of treating these degenerative diseases, but now scientists have found a connection between an amino acid and a reduced disposition to these diseases.
Ethnobotanist Dr. Paul Cox, who has earned degrees in botany, philosophy, and biology, has been studying the surprising connection between these two neurodegenerative diseases and flying bats in Guam. He believes that he may have found a new potential treatment for these diseases. So let's hear from the man himself, Dr. Paul Cox, who's coming to us live from Guam.
Hi, I first got interested in my current research when I was studying the Chamorro people in a region of Guam 16 years ago. I soon realized that this group of people were eating bats as part of their diet, and upon further research, I realized that the bats could be the cause of higher levels of neurodegenerative diseases observed in these people. I realized that big pharmaceutical companies were looking for cures for Alzheimer's only from one angle. The central hypothesis that was the basis of all the research was the amyloid hypothesis. This was centered around the fact that clumps of beta amyloid were causing the disease. The beta amyloid protein is formed from the breakdown of large proteins called precursor protein. Amyloid protein forms clumps that cluster between neurons and prevent proper functioning of the cell. Research conducted to target these clumps have led to tremendously expensive drugs that have had little effect, and I wanted to find an alternative option. After receiving funding from the 1985 Presidential Young Investigator Award and staying in Guam for several years, I decided to visit Samoa for a cure for cancer. Although this did not directly help my mission to cure neurodegenerative disease, I was successful in finding a natural tree bark that lessens the effect of HIV. By conducting field research, I was able to understand the culture of the Samoan people, which helped me find effective neurodegenerative disease and cure the epidemic. This led me to, I decided to keep this mindset when I learned of the major neurodegenerative disease epidemic, Litigo-Bodig, that was afflicting the Chamorro people of Guam. Many attempts had failed for corporations to find the cause of the high rate of the disease, but I looked at it from the lens of an ethnobotanist. I learned of the Chamorro culture, and I found that the flying foxes were the integral part of their diet. I talked to the people who considered the animal to be one of the most desired and sacred foods. Flying foxes eat cycad seeds, which contain beta-methylaminoalanine, which is an amino acid. Over time, the flying fox accumulates tremendously high concentrations of BMAAs. BMAAs are a neurotoxin which has links to several neurodegenerative disorders. There are currently 20 amino acids commonly used in making proteins. BMAA is an amino acid that is outside this group. It is a secondary amino acid compound in which one of the methyl hydrogens is replaced with a methyl amino group. It has been shown through lab experiments that exposure to BMAA has an excitotoxic effect in motor neuron cells by causing glutamate receptors to allow more calcium into cells, leading to cell death. Studies have also found that BMAA is mistakenly incorporated into proteins in place of L-serine amino acid during translation. When a protein takes up the BMAA instead of the L-serine amino acid, plaques and tangles form in the brain tissue as the protein deforms. To back my claims, in 2016, my lab and I conducted a trial on monkeys at the University of Miami's MASH. We fed vervet monkeys large amounts of BMAAs with and without the amino acid L-serine. The monkeys who ingested the BMAA without L-serine developed the plaques and tangles found in neurodegenerative patients. Monkeys who ingested the BMAA along with high concentrations of L-serine had 80 to 90% fewer tangles in their brain. As I mentioned before, BMAA takes the place of L-serine during protein synthesis, which may cause protein misfolding and plaque buildup. After conducting experiments on the vervet monkeys, it is obvious increased concentration of L-serine reduces the chance of neurodegenerative disorders from being onset. One reason may be that if we increase the concentration of the L-serine in the body, it will increase the probability of the tRNA synthetase to bind the L-serine and not the BMAA. Also, L-serine is essential for the formation of phosphatidylserine, which is a component of the membrane of brain cells. This was astounding, and with this new information that my team and I have uncovered, a cure for neurodegenerative disorders is finally a very real possibility in the near future. Thank you.
Thank you for sharing your findings, Dr. Cox. To help Dr. Cox further understand the inner workings of BMAA with lab work, he recruited microbiologist Dr. Rachel Dunlop. Now let's hear it from herself what they found.
With the working theory that Dr. Cox approached me with was that the BMAA, when bioaccumulated through natural processes, would be able to cause the symptoms seen in the elderly people of the Camaro group. He believed that the BMAA was passing as glutamine, one of the 20 vital amino acids required for cell function. My lab's experiments indicated that BMAA was not mimicking glutamine, but rather the amino acid L-serine. Through time and research, Cox discovered that their diet was filled with BMAAs due to the flying foxes that the older generations of people had eaten into extinction, as they are not very prevalent in their normal diet seen today. However, the discovery that L-serine was a neuroprotectant agent in the human body was a huge breakthrough in the field of neurology. The FDA has recently cleared L-serine as a safe dietary supplement, but Cox is working for L-serine to be taken by anyone who is potentially at risk for one or more neurodegenerative disease. L-serine can be found in normal diets. Foods high in L-serine include soy protein, peanuts, eggs, lima beans, and, luckily, bacon. In 2017, Cox published the results of a clinical trial of L-serine given in varying doses to ALS patients over the duration of 6 months. Out of the pool of these patients, the 4 that received the dosages of about 30 grams a day of L-serine saw the progress of their symptoms slow by 85% when compared with the other patients. The number of patients in this case, sadly, was too small for the finding to reach statistical significance, but if further clinical trials replicate anything close to that percentage, L-serine will slow the progress of symptoms far more than any existing drug, potentially allowing people afflicted with these horrible diseases many more years of life.
Thank you for taking time out of your day to explain your research and findings to us, Dr. Dunlop. Well, it seems as though we are no closer to curing Alzheimer's than we were 20 years ago, although Cox's trials and errors were in no way a waste.
Scientists from multiple different backgrounds and specialties were able to work on this project together, and they were able to combine their skills and get the job done. In the future, this may even provoke more scientists to cross the borders that define their own specialties, which wouldn't be a terrible thing. Discoveries in this area are still desperately needed.
In the absence of a cure, the pool of Alzheimer's patients will soar. While 47 million people worldwide live with Alzheimer's today, 141 million may have the disease in 2050, according to the Alzheimer's Association. We need a cure, or the number of people affected by these diseases will continue to grow. Hopefully, with more trials and research, L-serine could be used for more than just preventative measures.
Maybe. In addition to preventing the increase in Alzheimer's and ALS patients, L-serine could help people with other degenerative diseases as well. For example, muscular dystrophy is another degenerative disease that affects many people around the world. This disease is caused by a problem in the genes that control how the body keeps muscles healthy. L-serine, which corrects misfolding in proteins, could possibly help this issue. If these proteins are compromised, they won't be able to supply the muscle with sufficient nutrients. L-serine may be a possible treatment option for more than just Alzheimer's.
Well, I think that's all for tonight.
Have a nice night, and remember to eat lots of bacon.